This is a new subject with immune deficiency, which is of two types, primary and secondary. Primary result from genetic defect, which is either affect the adaptive or innate immunity and present since birth. An example of primary immune deficiency is skin disease, Digeorge syndrome, X-linked agammaglobulinemia, chronic granulomatous disease. First, we have the SCID, which is an abbreviation of the severe combined immune deficiency. It's an autosomal recessive. It is occurred with the deficiency of the adenosine deaminase enzyme, which is produced in the thymus, which is lead to defect in either T or T and B cells. SCID results in severe recurrent infections, is usually fatal, and it's incompatible with life until the baby put in the complete and isolated atmosphere, and the baby is more liable for the thrush, which is caused by the candida, which is called mammalia. And the second immune deficiency disease, which is the Digeorge syndrome, which is an autosomal dominant, which is a result from thymic aplegia, which is due to defect in the third and fourth pharyngeal pouch. It's resulted in cellular immune deficiency, which is affect mostly T cells, T helper, T cytotoxic. Is associated with deletion in the embryo of a region of the chromosome number 22. Is characterized by hypoparathyroidism and congenital heart disease, because they are originated from the third and fourth pharyngeal pouch. So this is the hypoparathyroidism. And the characteristic facial features that there is dysplasia of ears and mouth. This is normality in the ear and mouth, and abnormally long distance between the two eyes. Another disease, which is X-linked agammaglobulinemia, which is there is a defect in the B cell because there is absence of the immunoglobulin, and there is low level of the IgG, and also called proton hypogammaglobulinemia, according to the name of the patient's proton. And patients suffer from recurrent bacterial infection beginning at 9 months of age, because the baby is protected from the infection from the antibodies getting from his or her mother. So there is a defect in the proton tyrosine kinase enzyme, which is responsible for the maturation of progenitor B cell to precursor B cell and to immature B cell. So there is a defect in the proton tyrosine kinase enzyme. Another disease, which is chronic granulomatous disease, which is genetic disease. 70% of it is X-linked and 30% autosomal recessive. It's due to defect in the oxidative pathway, by which phagocytes generate H2O2 that kill phagocytosed bacteria. So this results in excessive inflammatory reaction, and here we can found by histopathology excessive infiltration the chronic inflammatory cells with or without abscess, and this leads to granuloma formation. And macrophage are sometimes involved in this granuloma. This is all the primary immune deficiency. The second type of immune deficiency, which is secondary immune deficiency, which is resulting from the chemotherapy like cyclosporine, azathioprine, corticosteroid in high dose, that's used in the organ transplantation. And the secondary immune deficiency can also result from the infection by virus like human immune deficiency virus, HIV-1, which causes a disease which is called AIDS, acquired immune deficiency syndrome. And this is the structure of the AIDS virus consists from the bilayer, and this is the nobs, and this is the core, and this is the RNA virus, and this is the reverse transcriptase enzyme. The replicative cycle of this virus entry of the HIV virus to the cell by binding of virion 120 glycoprotein envelope protein to CD4 on T helper cells, which is the important step, and then the interaction of the chemokine receptors CXCR4 and T cell tropic strain of HIV, while the another strain of the virus, which is its tropism to macrophage by the binding the chemokine receptor CCR5. The another step, the virion 41 glycoprotein leads fusion with the viral envelope with the cell membrane, and the virion enters the cell, then the uncoating occurs, and then the viral reverse transcriptase enzyme, which is the RNA-dependent DNA polymerase enzyme that synthesizes the DNA from the RNA. The viral DNA can integrate into the host cell DNA at different sites by the viral integrase enzyme, lead to formation proviral DNA. Thus, a person infected with HIV is considered to be infected for life because viral DNA is integrated into the host DNA. Host cell RNA polymerase transcribes viral DNA into messenger RNA and RNA genome. The messenger RNA transcribes to several large polyproteins, and then this polyprotein will be cleaved into small polyproteins by the protease enzyme of the virus, and then assembly into nucleocapsid and then virus bud from the cell. So each cell will be converted into a factory to produce large number of viruses. And this figure shows the life cycle of the virus. This virus will bind to the cell membrane and then uncoating, and then use the reverse transcriptase enzyme that convert the RNA to double-stranded DNA, and with the aid of the integrase, the virus enters the genome of the host by the integrase enzyme, and then use the host RNA, use the host enzyme to produce the RNA, and then synthesis of viral protein, and then assembly, and then budding of the virus from the cell. Transmission by sexual, vaginal, and anal blood transfusion, IV drug user, transplacental, adverse vaginal delivery by cesarean section lead to transmission of the virus, and breast milk and saliva, there is query of its transmission. People at risk: homosexual men, heterosexual, IV blood abuser, hemophilia, infected mother to her offspring, dentists, lab workers, healthcare personnel. HIV infect T helper CD4 cells and kill them, leading to immune suppression and increased infection with opportunistic pathogen. And immune suppression leads to development of cancer like Kaposi sarcoma and lymphoma. So, and there is also increased infection with the pneumocystic carinae in AIDS patients, which is lead to diarrhea. And there is a small bowel AIDS infection with cryptosporidium. Initial infection of the genital tract occurs in the dendritic cells that line the mucosal Langerhans cells, and this local CD4 T helper cells will be infected, lead to decrease in its number. This cytotoxic CD8 will be attacked, and leading to death of T helper cell and the HIV-infected monocytes and macrophage, producing multinucleate giant cells. HIV acts as a super antigen, lead to increased synthesis, leading to activation many T helper cells, and antibody also appears in the serum of the patients against the GAG protein 24 within 6 to 12 weeks. So after exposure to the virus, and this takes 6 months to increase, and this is detected by the ELISA. And then the antibody against the glycoprotein 24 will be detected by the test Western blot. So at the initial infection with the X virus, there is no increase in the antibody unless after 6 to 12 weeks. T helper 17s will also express CCR5 and CXR4 chemokine receptors. ADCC also from viral infected cells, HIV infected T cell, HIV induced T cell apoptosis. HIV leads to polyclonal activation of the T cell, and some HIV infected individuals are long-term non-progressor because they produce large amounts of alpha-defensine that interfere with HIV binding to CXCR4 receptor and block entry of the virus into the cells. All these lead to immune suppression. Also, the HIV virus have a mechanism to evade the immune system. First, by the integration of the viral genome to the host cell genome. Second, high rate of mutation in the envelope gene, production of TAT and proteins that downregulates the class 1 MHC proteins required for ET cytotoxic.